So friends, today we are going to discuss about gene interaction. This is BSc third year Botany chapter, that is gene interaction. I introduce myself. I am Dr. G.V. Honna, Department of Botany, Sri Siddheshwar Mahavidyalaya, Mazalgaon. So friends, today we are going to discuss on gene interaction. The first, that is the definition of this gene interaction. Particularly, the definition of this gene interaction, the phenomenon of two or more genes affecting the expression of each other in various ways in the development of a single character of an organism is known as gene interaction, or it is also known as interaction of gene. मराठीमध्ये सांगायचं झालं तर जीन्स जे असतात आणि जे आपोआप किंवा आपापसामध्ये कसे बिहेवियर करतात, वर्तन करतात, त्यास इंटरॅक्शन ऑफ जीन म्हणतात. ते कधी आपल्या आपापसामध्ये करतात किंवा शेजारील असलेल्या जीन्समध्ये सुद्धा अशी वर्तन करतात, त्यांना इंटरॅक्शन ऑफ जीन असं म्हणतात. मोस्ट ऑफ द कॅरेक्टर्स ऑफ लिव्हिंग ऑर्गॅनिझम्स आर कंट्रोल्ड or influenced or governed by a collaboration of several different genes. Mendel and other workers assumed that characters are governed by single genes, but later it was discovered that many characters are governed by two or more genes. So genes affect the development of concerned characters in various ways. This lead to the modifications of the typical dihybrid ratio 9:3:3:1 or trihybrid 27:9:9:9:3:3:3:1. Then the gene interaction is of two types. According to Batson and Punnett hypothesis, some characters are proposed by the interaction of two or more pair of genes. That's why there are two types. The first is intragenic. It is also known as allelic gene interaction. And another is intergenic, or it is also known as non-allelic gene interaction. Then the first, intragenic or allelic gene interaction, when two alleles of a gene interact in such a way to produce a phenotype differ from typical dominant recessive phenotype. Example of intragenic: incomplete dominance, codominance, and multiple alleles. Then the second type, that is intergenic, or it is also known as non-allelic interaction, when two or more independent genes present on the same or different chromosome interact to produce two different expressions. The example are epistasis, duplicate gene, lethal gene, complementary gene, and supplementary gene. Again, the next, first we will see today, that is intergenic or non-allelic gene interaction. The first is the complementary gene. The ratio of this complementary gene is 9:7. If both genes loci have homozygous alleles, both of them produce identical phenotype in F2 ratio become 9:7 instead of 9:3:3:1. The definition is also two or more non-allelic genes interact with one another to produce a character, but one gene cannot produce that character in absence of other. In such cases, the genotype c, capital A, small a, capital C, capital A, small c, small a, capital C, small c produce one phenotype. Both dominant alleles, when present together each other, are called complementary genes and produce a different phenotype. The Batson and Punnett observed that when two white colored flowers, that sweet pea variety, the scientific name is Lathratus odoratus, these two plants are crossed. F1 progeny has produced, that is red colored flower. When this F1 progeny red colored flower was selfed, then F2 ratio or offsprings are formed, that offsprings are in 9:7 ratio. The 9 colored flower are red and 7s are the white. Then the explanation of that, there are two varieties of pea plants, one is red colored and other is white colored. The red colored of flower is due to anthocyanin pigment. The anthocyanin is produced from a colorless substance called chromogen by the action of enzyme or activator. The chromogen cannot be converted into anthocyanin in the absence of the enzyme. Thus, the production of red color, both chromogen and enzyme should be present in the plant. In absence of anthocyanin and the enzyme, red color cannot be produced. A dominant gene, capital C, is responsible for production of chromogen. When the gene is in recessive form, that is in small c, the chromogen cannot be produced. Similarly, another dominant gene, capital A, is responsible for the production of enzyme which convert the chromogen into anthocyanin. When this gene is recessive in the form of small a, the enzyme cannot be produced. Thus, chromogen cannot be converted into anthocyanin. That's why the capital C gene produced chromogen and the capital A gene produced the enzyme. When this capital C and capital A, both genes are present to produce the anthocyanin pigment, and the flower color comes in the form of red. That's why red color is produced by the interaction of both dominant genes, that is capital C or 1 or 2, capital A 1 or 2, or only capital C and capital A also present, that forms the flower color is red. This table shows the explanation of that is capital C, small a, this type of genotype, it is the white colored flower. And another plant, that is small e, C, capital A, that is also white colored flower. Both are crossed to form the new progeny, that is capital C, capital A, small a, that is red colored flower. This is the form of F1 generation and it crossed selfly. Then the new progeny is formed. In one red colored, another red color forms the 4 types of gametes, and that 4 types of gametes are crossed to form 16 offspring. Out of 16 offsprings, the 7 are the white colored and 9 are the red colored flower. That's why this explanation shows the 9 colored flowers are formed from this crossing, and 7 colored or the 7 are the white colored flower. Means the both type of genes, that is capital C and capital A, when they are present to form the red color, the anthocyanin pigment, this type of gene interaction is called as the complementary gene, and the ratio is 9:7. This is the best example in the complementary gene, that is Latheratus odoratus. Then stop here. Thank you.